the last talk of the day. Christy Hipsley, here from University of Copenhagen Biology Department, with visualizing evolution in 4 dimensions using X-ray CT, and you also promised here.
Yeah, you might have.
But I'll leave the time to you.
I did so. Yeah. For anyone on Zoom or that I haven't told personally or this morning, I did change my talk subject last minute, and that's because I felt the canid work that I'm a part of, like wolf dog evolution, is still very much a work in progress, and so I wanted to kind of present it to our internal group to get feedback, which I did, and that was really great. So thanks for that. So I'm kind of defaulting to a talk I've given in various forms, maybe in the past year or so. So if you've seen it, like log out, take a nap, or whatever you want to do, but it's basically just kind of looking at the many ways that digitization benefits evolutionary research, and really with a focus on museum collections, because that's my background. And this is just a very nice visualization. It's not mine, but you might recognize it from Landmark Editor. That was kind of one of these, I think the first like open access 3D landmarking software, which now is Stradivan Checkpoint by Dave Wiley at UC Davis. So, but it kind of sums up the things that we want to use digitization for. So we have 3 dimensions, and then we can translate that into evolutionary time. What direction do I? There we go. Okay. Yeah, so I've heard kind of digitization, it's called a revolution, or the resolution revolution, I've also heard. And there's lots of reasons why it's extremely beneficial, right? So I'm not talking about computed tomography, CT, but which can take several forms, but I'm basically, for my purposes, going to focus on X-ray CT today. And we all kind of know the pros of this, right? It's non-destructive. You get the same resolution in all orientations, which you don't with, for example, microscopy. And then you can spin this thing around, right? You can rotate it to look at different angles. But there's still challenges, and we've kind of touched on a lot of these, and you probably know. It still involves some sample preparation, and if you work on dead, like floppy, wet things, that preparation becomes really, really important. These things have to stay still while they're being digitized. It's a big jump above what we used to do, which was something like serial grinding, you know, and museums don't really like when you cut things apart. So again, there's a big benefit. But if you had something like fossil, you know, you'd grind a layer, take an image, grind a layer, take an image, or maybe you'd put these things in between glass slides or something. Tissue recognition, you know, for X-rays to work, they have to be attenuated, and soft tissue's not very good for that. I'll get back to that. Post-processing, I would say this is getting better all the time, and of course, working with like machine learning and stuff can help. But I would say that the biggest challenge we still have is large sample sizes. So I'll talk about that. But we're getting really good at it, right? So not only does it give you really awesome 3D images like we've all seen in the past days, but we can extract a lot of information. So this is the same specimen. This is from Ed Stanley at the Florida Museum, and who's a big contributor to Morphosource, and the OVERT project to scan 20,000 animals and make the digital data free. It's really available. But, you know, we can look at diet, we can look at parasites and reproduction and all kinds of stuff. And we're starting to look at soft tissue. You can trick an X-ray into thinking, you know, it's into attenuating soft tissue by basically bathing it in some kind of a metallic solution, right? So that the tissue becomes more dense. So I think we're getting better at all of these things. And we're still, you know, we're using this a lot in natural history museums. Most of us are taking dead things from museum specimens and we're scanning them, and because they have lots of other information that we want to then ask, you know, kind of evolutionary questions about. And so I'm going to just take the time to go through some of the examples from my career of different ways that I've used it, and they're not all morphometrics. There are lots of other things you can do with digital data, right? So species descriptions are kind of a really basic one. looking at inter and intra-specific variation. When you know that, you can maybe map it onto a phylogenetic tree to look at evolutionary processes. We've heard about that already today. When you have the right kind of specimens, you can look at reproduction or development. Sometimes we get lucky enough to have those in museums. This is becoming a really big one nowadays, having cyber types or even these whole eye collections, what the Danish Natural History Museum and many others are doing, and just loads of other downstream analyses. So it's, it's a very flexible tool. So I'm just going to run through some of these that have been fun in my career. And a lot of these were from when I was I did a PhD and postdoc at the Berlin Naturkundemuseum, and I also worked as a CT technician. So I just got to scan lots of cool things and get involved in lots of projects. So basic species description, this is, if you've ever been to the Berlin Naturkundemuseum, you have the big dinosaur hall with the famous Tenderu dinosaur group there. Jurassic dinosaurs, this is the Titillators, an herbivorous dinosaur. And so it's really good for looking at these kind of animals, which are maybe fossilized, rare, or some cases small. And you can also not only describe something in depth, but kind of extend that into try to understand what it did in life. So here we did we CT scanned the brain case, not the whole thing. It's obviously a big animal, but just a little part of the inner ear and the dorsal skull. And so here is actually a case where we were not as much interested in the bone, but the empty space within the bone. So those are where the ear canals are that carry sound. And from that, we could reconstruct things about its posture. So remember, you used to have dinosaur pictures that the Brachiosaurus standing with its head straight up and its tail straight down. That's probably not the case, right? So we can see it probably had a nose up posture. We can even estimate its hearing range and compare that to other living forms. So, so that was kind of a fun project to be part of. This is my darlings. I love reptiles, mostly lizards have been the thing I've worked on. And this is a group of skinks, but they're not snakes, but they're limbless or in various degrees of limb reduction, but they're like as thin as a pencil, basically. They're really hard to dissect, right, and look at the bones. So it's again another case where CT becomes really important, and a lot of these are rare. We only maybe have found one. They live underground, might I add, so they're not easy to find, right? So this was a whole group of skinks from Madagascar. We could CT scan them and look at the girdles, so everything from no limbs to fully limbless. And then we can ask things about like the timing and rates of these changes. So again, like people have presented today. And so we can map that onto molecular phylogenies, look at when does limb loss start to occur, what's going on in her Earth history at the time. Do we have changes in environment that would make this beneficial? And this idea of intermediate forms, we think, do you just go down a black hole of limb reduction? But these things actually seem to stick around stably for long periods of time. So there's probably functional benefits to having little flappy arms. And we can test some evolutionary hypotheses with this. What is like Dolo's law, the idea that once complex traits are lost, they are not regained. So reversals, basically. Here we don't see, you don't tend to regain limbs once you've lost them, but in other groups, and also thanks to CT data, we do find that these things are happening. So some like teeth and frogs. Reproduction, this was a really fun project to work on. Invertebrates, so this is a bush cricket, and they have these genital features called Titillators. So that's if you pull back some of the tissue and expose them. And males have them, and they didn't know what it was for. Well, the name maybe tells you it's something involved in reproduction. And so, sorry for these poor crickets. They were in a colony, and while they were copulating, they were flash frozen. At least they would die happy, I hope so. And so then we CT scan them. So this is the female, she's full of eggs. That's what all these structures are. That's the male, and then in yellow are his paired Titillators. And we can see from this that they are percussing against the female's reproductive plates. And then we combine that with SEM of those plates. I mean, obviously these things are tiny. And we can see indeed those are covered in these sensory receptors. So they are titillating female. But they didn't know that actually, so yeah, I guess it was a good guess. But this was a really fun project to work on for me. And obviously, you know, this has big implications for things like sexual selection and how these structures evolve. Developmental biology, I think, has huge potential in this field. Again, if you have the right collections, so you know, support museums. This was a really famous collection of humpback whale embryos. How do you get an humpback whale embryo? I mean, the smallest one was this big, you know. So, and then the biggest here was maybe that big. But they all came from Norway, actually, around like late 1800s. So, yeah. Luckily, someone has kept these around for that. Obviously, they're not going to let us cut these up, but they're really important to know if you want to know about whale development and by extension evolution. So I'll just show two of them, for example, the CT scans. There was some weird preservation stuff back then. Who knows? People sometimes put it in a bottle of vodka or something, you know, if they didn't have anything. But a cool thing you can see, for example, is in this extremely early stage. So humpback whales are baleen whales, right? They have these cones of baleen, but at this very early stage, we actually see in the jaws, they have teeth alveoli, so the sockets of the teeth, and then they disappeared. But if we didn't have this tiny snapshot, you know, we wouldn't know that. We can then do things like, yeah, did you see that? I'm sorry, my computer's a bit slow. I see on my screen, but not there. Ah, wait, okay. Yeah, you can do things like order the timing of appearance of certain traits, developments of certain traits. So the exotympanic, so part of the inner ear, evolves or appears very early in whales compared to terrestrial mammals like a mouse or a pig or a human. Underwater hearing, of course, is really different and really important for whales. So that is maybe not surprising, but it's something we can document. We can look at evolutionary relationships. So whales and cetaceans, you know, are evolved from kind of ungulates ancestors, and we can then compare, you know, something like a dolphin inner ear to a fossilized inner ear of one of these ancestors, and look at similarities and look at differences. Something that many others of you do. And I think this is a really promising and really exciting area that I would like to get into more. I'm not a geneticist or genomicist, and I don't plan on becoming one, but I hope to work with people who are. So I think, again, KU and where I'm at is a really good place to do that. So one way we have been looking at it, I spent 5 years in Australia. Lots of lizards, it's great. Not so many here in Denmark, but many of these lizards are cryptic, what we call. So you can't, even for someone as a trained expert, if you pick them up, they're very hard to distinguish, a lot like the butterflies that Nikolai talked about earlier. And so can we use CT scanning combined with geometric morphometrics to delineate these cryptic species, to tell them apart? We have molecular phylogenies for them, so mitochondrial DNA, nuclear DNA tells us that these are different lineages, but again, they just look so similar. In this case, we focused on skulls. We kind of like skulls for lots of reasons, but they contain lots of traits and information, but they hold, you know, they interact with environments, they hold sensory systems, they're very ecologically important. So they're good things to work on. And it might be hard to see any of the differences in these shapes, but they are different. And that's, again, the power of geometric morphometrics is, you know, I say usually you find evolution where you look for it, but in this case, your eyes will never see these differences. But right when we map this onto, so this is a morphospace based on landmarks, and then we project the phylogeny into that space, and we actually see, okay, we're seeing differences. The skull shape variation is kind of mirroring the differences of the phylogenetic distances in these groups. And, you know, in some of these, it may be incipient speciation, and some maybe, you know, they're forming real species. And in this, and also combined with many other forms of information, what we are calling an integrative taxonomic approach, from that, we identified 5 new species and also multiple hybrid zones. One thing that I really also love is convergent evolution. So we've mentioned it several times today. And that's also because I have worked on reptiles and squamates where it's rampant, but mammals do it too. I always say I'm not a mammal person. I mean, I'm a mammal and a person, but I've never worked on mammals. But I thought, okay, if I'm going to work on mammals, I'm going to work on marsupials. And if I'm going to work on marsupials, I'm going to work on a carnivorous marsupial. And I got to do that in Australia. So you might recognize this is the Tasmanian tiger or thylacine, a stripy marsupial, right? And it's very famous for being convergent with wolves or canids. And so this is something we would like to look at in a morphometric and evolutionary sense. However, this animal is extinct, despite that every year, like a Dutch or German tourist sees one, never gets a good photo, but some people claim they're there. I'm sorry to say they are extinct, but we do have dead animals, dead specimens of them in museums. We have, skin, skulls, skeletons and stuff. So we could scan, I don't know how many we got in the end. Yeah, several of a dozen or so of the thylacines, lots of other mammals. We also got data from Anjali that had collected the same landmarks over fossil mammals. And we could actually, we could do lots of different things. This is just like a smattering of work from different papers over the years. But we first started out just saying, yeah, they look similar. We just eye that up and we say, yes, they're convergent, but like, can we quantify it and how? And putting morphology into a geometric context lets us do that. And so where we actually, so this is again, one of these morphospaces, you can see this is the Marsupial Morphospace, this is the placental Morphospace, and thylacine is the only marsupial really that crosses into that very canid dominated morphospace. This is another one, a really stem marsupial. That's one of these like big saber tooth kind of guys. It's convergent with the saber tooth cat. And we could verify statistically that this is highly correlated with diet, carnivory in this case, not surprisingly, but still we can put numbers on this. And we can even estimate the amount of evolutionary kind of phylomorphospace that has been closed over time between these convergent lineages. And that's not just looking at PC1 versus PC2, but we do this over the entire kind of variation of skull shapes in this case. That was our package called Conv Evolve by Tristan Staten, if anyone knows it. Really cool package. We then went on to look at development. So remember, these are extinct animals. They're marsupials. So sometimes when a marsupial dies, it has babies in its pouch. And in some of these few cases, when they were kind of extirpated in the 1800s, early 1900s, some of these pouch young came into museum collections. And there were 13 known in the whole world. We worked very hard, had to make lots of museum agreements and so on to get them. And we managed to CT scan all of them. So these are just the main, some of them were siblings, but here are basically the main stages, growth stages that we sampled. Except this one, you might notice it looks a little bit funky. It was labeled as a thylacine. And from the CT data, we could tell that, it has different number of vertebrae. Mammals don't do that. Like, we're very conservative with our vertebral numbers. And it also had, some other different features, like very well-developed epipubic bones that many marsupials have. And the thylacines are very small. So from this, we can say, actually, we think it's a Dunart. Sorry, museum, and sorry, us, the number just went from 13 to 11 pouch young specimens known in the world. That was not our point. We actually wanted to look at development of forelimb versus hind limb, getting into some different marsupial developmental biology, but that was one of the outcomes that happened from these type of technology using CT. Another thing we did, very inspired by Anjali's work, was look at integration of different modules, cranial units over time. So, right, you don't just have a nose and an eye and an ear and stuff. I think I pointed wrong, but these things work together, and they might work together in different ways over different times of the animal's life. And so we could, in this case, landmark the skull more densely, and then look at patterns of integration over time. So we compared thylacine and wolf and tried to increase our specimen sampling. And here we also look at differences in mesoderm versus neural crest cells and how that affects those patterns. So that was a really nice study. And finally, what we really want to do is get into more genome-phenome mapping. And we can do this because this one very special pouchium specimen that has been in a jar of ethanol for over 100 years gave DNA. It gave good enough DNA that we could sequence the whole genome. I mean, it's not super complete, but good enough to do these kind of analyses. And so this gives us, it's the most complete genome of an extinct animal, I think, today. Maybe passenger pigeon was published like the same week our paper came out, so that was a little bit of a shadow, but good for them. Yeah, so now we have these genomes, and we can look at now what genes or what regions are in common between these two very distantly related organisms, a thylacine and a wolf, that look so similar, but they've last shared a common ancestor like 160 million years ago. So we can start to try to, in an extinct organism, say which genes or gene regions. And here we actually see that they're regulatory elements. So they're not actually the protein coding genes, but it's the things that tell them when and where to turn on and off. And we've identified, my colleagues, Andrew Pass group at the University of Melbourne, identified that 339 positively selected in both of these convergent lineages, and many of them are indeed enriched for functions of the skull that are related to eating meat, for example. So that's been a nice project that also keeps on giving. And this is a great thing that we've just done come out. So we want to continue on this thylacine work and the genome-phenome interactions. You actually need models to do this, right? And for marsupials, we have the North American, opossum, can be considered a model, but it's actually quite distantly related to Australian marsupials. So it doesn't really help us when we want to look at kind of development and genetic, mechanisms. So we have just published a paper introducing a new marsupial model for Australian marsupials, especially the fat-tailed dunnart. It's a little marsupial mouse. And this is just an example that at birth, and remember, these are marsupials with very different reproduction, this would be the size difference between, a mouse at birth and a dunnart. So they're little bitty baby things, but they're kind of just like hamsters. You put them in a cage and give them pellets and a running wheel and happy. And the mother has babies in the pouch, and you, not me, another student, Laura Cook, could sample them at exact known time points every day. And you don't have to kill the mother to do that, right? So that's a really great resource. And we CT scanned every single one of those specimens to also give complementary morphological data in 3 dimensions. We've made those open available. You don't even have to ask. You can just download them from Morphosource. And these also include tissue stained samples. So this is, there you saw the eye. Yeah, I think now you see it's a little baby like this. So we can even look at soft tissue and organ development, and use this as a model. Again, maybe now to compare to some of these thylacine pouchium that we have. So I just want to end, I'll take a couple minutes, that I still think the outstanding challenge, and we've kind of danced around this, I think, over the course of a few days, is large sample sizes. So if we just look at the number of scientific articles in the past decades that include CT, we see that, so the top one is biomedicine. Obviously, that's been going on for a while. biology, at large, that's excluding like veterinary and human medicine. But ecology is still really down there. We're still not kind of translating those efforts into asking really ecological questions. And I think we have the power to do that. We have those sample sizes, right? You all been to museums. This was the Melbourne Museum, why I have this image and not the museum I am next to now, but we've got lots of specimens. So not only these kind of stuff, big stuff on display, but for especially those of you who work in museums or who went on our tour yesterday, that most of those buildings are just filled with rooms and jars of this kind of stuff, dead things that can be digitized. Some of them are big and some are really, really small. I work on reptiles again, or amphibians, they're really tiny. So CT is great for that. And these really number in the billions. So this is just a huge resource to tell us about kind of the spatiotemporal patterns of life. I've started to kind of try to tackle this. We need high throughput approaches, of course, coupled with things like machine learning, big data, big computers, and so on. I tried to tackle this in fossils. So these are fossils from Queensland in tropical northeastern Australia, where we have cave deposits just containing, like, you could bathe in the number of fossils that are from here of little tiny lizard jaws and frog ilium, the hip bone. These are really diagnostic. They're super interesting for lots of reasons, but there are a lot of them. They're just so small. How can we dream to study these in some kind of community level way? And so we kind of have come up with, it's kind of almost embarrassingly simple, but you still have to pack these specimens in some systematic way where you can then go back and identify who is who, and link them to their own data, right, where they came from, and so on. And then we of course want to use those for analyze those using things like morphometrics, and where automated or semi-automated approaches are just necessary, because like Nikolai said, you don't want to just be clicking on these thousands and thousands of specimens. We're now trying to tackle this high throughput CT and post-processing together with the Danish Technical University or DTU, and for things that are more medium size. If you have very small, well, maybe we can use neutron scanning or synchrotron. But for these and big things, well, you're probably going to do some surface scanning. These medium-sized objects, we have a lot of them, right? So how can we deal with them? We probably need some robots to help us, right? So when they told me that, oh my God, robot, I thought, oh, it's going to be something like this. It's probably something more like this. Just like it's a robotic arm that we are working now with the museum to help select collections that would be most amenable to this, pack them in some standardized way, and then we have a robotic arm that can move them in and out of the machine overnight, over the weekend, right? So the scanning is not the hard part, but you just need the power and the movement to get these things through the system. So a really good example of a nice collection for this would be the polar bear skull collection. So these have been systematically collected, hunted over the past decades, because they are looking at traces of pollutants and stuff in the Arctic. So they've seen, for example, changes in bone mineral density related to pollutants and stuff. So, this might be a nice collection to do that with. But, we're going to end up with a lot of data. So I'll just end on kind of what I propose. And if anyone wants to help write some grants together with me, this would be great, for some infrastructure here to build what I call the high vis lab. So yeah, a visualization facility for high throughput phenotyping. And I think this could be model organisms, you have mouse colony, for cancer research, or it could be polar bear skulls or whatever, that you can scan or image in some way. So this is kind of what I would, my goal to get money for, things like, it basically just needs to be a big computer lab, and that's equipped with software. It would be great to continually have an updated library of codes, things that you guys like, that Mike is developing in R and Geomorph, and literature, procedures. yeah, it'd be great to have some IT support because this is big data issues, right? And Morphosource people know that very well. You need all kind of data storage, backup, blah, blah, blah. I'd like it to be here where I work. So that's my proposal. But it makes sense, right? This is biology, here's the zoology museum, here's Diku, my partners in the Zoom Synergy group, and DTU is kind of up the road. So that would be the goal. Oh, and yeah, I'll just go through this quickly, that let's have things like Anjali's method. Let's have that like baked into the system. She's given a list of everything we need, workflows, you know, it's there. We don't only have to do landmarks, right? We can do these kind of what Doug talked about yesterday. Yeah, we can do kind of non-landmark approaches that are semi-automated, and there's other ones as well. Oh, sorry. So yeah, I would like to equip this lab with these kind of things, give people options, right? And then maybe we can start to contribute to really kind of community level analyses of phenotypic change now in the Anthropocene when we really need it. Yeah, I'm going to end with that. Maybe if my I can see it on mine. Okay. There we go. Yay! Okay. This is a little fellow. Yeah, I don't list all the people I thank, but it's obviously all the museums where I've worked over the years, all the curators, all the managers, all the people that help collect the data. Working with the group I'm with now is really exciting because I think we're going to do lots of things that I've never thought about, but you know, I do have lots of data. So, and I know other people that have lots of data. So I think that was a kind of a goal with the workshop now, to identify those synergies. And yeah, this is just again one of these thylacine pouch that we are able to look at even organs and stuff, and this is very beautiful. So thanks, and yeah, get in touch if any of this like sounds at all interesting to you. Although I already kind of know everyone in this room, so yeah. But anyway, thanks a lot.
So, questions?
Yes, me? Yeah, so because it's great. Usually tropics aren't really great for fossils. You might know things like decay quite quickly, but there's cave systems, huge limestone cave systems, and most of these little lizards and little mammals and frogs and stuff were probably brought in by bats and owls. And so they either puke or shit out the bones, and there they are for us. So they're in very, very high resolution, and they've been really well systematically sampled at like the centimeter levels. And I have those for various deposits for the entire Cenozoic, like going back about 50 million years. And so over that time, Australia has gone through wet, dry cycles. They had contact with New Guinea, you know, when sea levels were low, then not again. So you also could have species coming from that direction or going that direction. So it's a super dynamic system. And lucky for us, those things that are in those caves are really probably the frog ilia more than even the lizards. I hate to say because I love the lizards, but they're probably very telling or very helpful to reconstruct those changes also at a community level. So I'm not just saying, oh, the species is there or it's not, it disappears, or, you know, but at that level, I can actually look at micro evolution. So it's not, is it there or not, but it's how is it changing over time. That's what I want to do, but that's like, I can't do it like alone, you know. I don't expect the students going to landmark thousands and thousands of things. So machine learning or anyone that wants to help me would be great.
Anybody else? And thanks again.